Hello, my name is Stephanie Besoiu, a fourth-year biomedical science student here to discuss my summer's research and honors thesis research project. All of you are very well aware and acquainted with SARS-CoV-2, which has caused over 200 million global cases of COVID-19 and over 5 million deaths, but overall an insurmountable toll on our daily life. This single-stranded RNA virus has a genome which consists of 14 non-structural proteins, these mostly involved in viral replication, and 4 structural proteins, involved in the physical structure of the virus. So this includes the nucleocapsid, envelope, membrane, and spike proteins. Most current mRNA and viral vector vaccines target this spike protein as it mediates viral entry into host cells. However, as shown by the emergence of variants, the S protein is mutation prone, potentially undermining the effectiveness of current vaccines. While mRNA vaccines appear to be effective now in preventing SARS-CoV-2 infection, it is unclear how long-lasting the cellular immunity is, as in how well do these vaccines activate T cells which kill infected cells. The extent of which these cells are activated has previously shown to be an indication of the length of protection given by a vaccine. This necessitates the need to evaluate other novel vaccine targets that may induce long-lasting T cell immunity, such as the other structural proteins. Therefore, this is the overarching goal of my project, which is to create and evaluate the immune response to vaccine constructs containing the promising vaccine targets of the nucleocapsid, envelope, membrane, and spike proteins. This is where SNREP5 comes in. It's a novel alphavirus DNA vector that contains the genes for one of our SARS-CoV-2 proteins, as well as proteins required for replicating the genome of the virus, so the non-structural proteins, but not any proteins required to actually assemble the virus. So what this means is when we add SNREP to another DNA called DHBB that contains the SNREP5 structural proteins into cells, into baby hamster kidney cells, we get virus-like particles, or VLPs, which contain the protein of interest but cannot spread and only have one round of infection, making them safe for vaccine use. We can take these VLPs and vaccinate mice with them, where there we can evaluate the antibodies the mice form to each vaccine and study how well do the mice T cells recognize and kill cells expressing one of our structural proteins, overall showing how well does the immune system respond to the vaccine. So to summarize my main objectives, firstly, it is to separately clone the structural proteins into SNREP5, and then secondly, put these constructs into cells that package them into VLPs, the vaccine carriers of the protein. And then lastly, I will vaccinate mice with these VLPs to evaluate the immune response to each construct. So far, the E and N expression constructs have been created, as well as an N and EGFP expressing construct. So EGFP is a green fluorescent protein that allows for visualization of BHK cells that are making VLPs under a fluorescent microscope. So these constructs have been packaged to VLPs in cells. The other constructs containing the other structural proteins are still currently being optimized. So these fluorescent images show that the N and EGFP expression system produces high amounts of VLPs. In the image on the right, the green dots show cells that have packaged VLPs in comparison to the general amount of cells present. This result is also present for VLPs containing just the N protein. So now looking at our structural protein expression in cells using Western blots, the image on the right with the nucleocapsid protein shows that BHK cells are expressing the N protein, meaning that the VLPs are working and are effective in mammalian cells, while the image on the right shows the predicted protein band size for the E protein. This is currently being optimized. So what these results demonstrate is that this system generates virus-like particles that could be used as a vaccine. So the future directions of this work is to continue optimizing the generation of VLPs for all of our SARS-CoV-2 structural proteins, and then eventually inject our VLPs into mice and evaluate the immune response, both of the antibodies that are formed and the T cell immune response. We could also potentially expand this platform to vaccines expressing multiple proteins at once, for instance, the N protein and the S protein simultaneously. But development of this novel viral platform will be another tool for tackling future outbreaks of SARS-CoV-2 variants, as well as other infectious diseases, speeding up the process of vaccine creation. So I'd like to thank my PI, Dr. Guido Van Marl, as well as Michelle and Rupali for their continued help on this project, and all the lab associates listed here. Thank you for listening.